So hello everybody and welcome to another session of All In with Bulpin. And today I'm joined by Heide Webster and Marc Gaal, who are joining us from our Carlsbad facility in California. We're going to have a chat today about cell and gene therapies. Heide and Marc have been involved in this space with the company for over 25 years. So welcome.
Thanks
you for having us. My pleasure, our pleasure to be here.
You really are both true pioneers in the cell and gene therapy space. You've been on this journey since 1995 with our company. What are some of the major advances you've witnessed over that time?
Well, we've come a long way in this space. I mean, there's a lot of things that I'm proud of, but a couple that come to mind are in that time we've produced over 500 batches of virus, and 3 of those particular products have gone on to be approved by both the FDA and the EMA. So those are some out of the gate kind of accomplishments that the site has had. The other thing that I'm proud of in most recently is the completion of a $110 million facility that we have here in Carlsbad, California. It's huge. It's behind both Heide and I right now, and it has the ability to scale up and produce large quantities of virus to the likes that we've not seen before, Heide and I, in our time. So I'm real proud of that. And lastly, I'm really proud of how far our customers have come with some of these drugs. There was a lot of promise early on in the industry and a lot of learnings that took place. But, now the conversations that we have with our clients, they're not about how do I make, one batch of virus to meet my milestone in clinical manufacturing. They come to us asking how we can get to commercialization, how we actually reach the finish line, which is getting these potential cures to the patient. So those 3, I guess I had an extra one there, but those 3 are kind of a sum of past, present, and future that are really kind of indicative of where we've come in this journey.
For me, we started out on the University of California, San Diego School of Medicine campus in some very small laboratories and very small clean room areas, producing very early gene therapies. We were one of the very first to try to do this type of therapy under GMP conditions. And the industry has developed quite a bit because in the early days, gene therapy was very exciting and seemed to be very effective or had a lot of promise, but it wasn't very safe. And patients were having some severe side effects around immune response, and just the selection of the pairing the patient with the therapy wasn't always going well. Now it's even more exciting because not only is it effective, but it's safe, and we're able to put these products on the market. And larger patient populations are seeing the impact.
Yeah. Okay. So thank you, guys. Having said that, although it's been around for several decades, cell and gene therapy is definitely still in its infancy as a modality. So what do you see as some of the biggest hurdles today when you're dealing with our cell and gene therapy customers?
Well, it's still quite a hurdle for our customers, and what I mean by this is process development and the timeline. So there is a need to manufacture these products safely, as Heide just mentioned, but there's also some competition to bring these to the market quickly, and so everybody wants compressed timelines. Typically about 3 to 5 years is what the expectation is to bring these products to market. But that's very challenging in an environment where there isn't a standard production process template. So that doesn't exist for the gene therapy space as it is today, but we are working diligently to complete that process.
Okay.
I would say the speed is definitely an issue in the regulatory side of things. Most of our clients are fast-tracked We are making things GMP very quickly right out of research tech transfer, and we're running GMP manufacturing for them. The regulatory agencies themselves aren't completely up to speed with all of what they should be requiring. They're still learning as we're learning. So it's definitely a challenge. The traditional clinical path isn't always going to work for these therapies. And certainly the smaller companies need a lot of hand-holding and assistance in getting their products into the clinic and then into the market.
Got it. So it's very clear that you guys understand what the challenges are and where we can add value for our clients. What do you see next on the horizon for cell and gene therapy?
Well, a couple of things. I mean, first, most of today's approved gene therapies, the three that we mentioned earlier, plus a few others, they are really targeted towards very small patient populations, rare disease indication, things like that. In the future, in order to democratize some of these, we're going to have to be able to scale up and produce larger amounts to accommodate the disease landscape as it changes. And this involves much larger patient populations to treat some more common indications like glioblastoma or non-Hodgkin's lymphoma, sickle cell anemia, you name it. There's roughly 7,000 genetic diseases that this particular therapy has the potential to cure. So there's lots of future in this, but in order to achieve the dream, we've got to be able to make more and scale up. The ways that we're trying to achieve that is a couple of fold. One is scaling up in viral vector manufacturing. I mentioned the facility that we've just recently finished building and bringing online. 88% of the gene therapies in development right now leverage viral vectors as their delivery vehicle. So we're going to need to scale up in order to meet that demand. The other thing is it's awful difficult when you say viral manufacturing. Not all viruses are created equal. They purify differently, they produce differently, they have different purity requirements from one to the next. So the way to maintain or increase the speed that we need while maintaining the quality standards that we have in our organization, we're going to need to use our templated process called Virus Express. It is a production process for lentivirus, and it involves a chemically defined media and an associated fully characterized cell bank. And that in itself will, and it's produced in suspension, that will allow us to produce much larger quantities with less variability, more consistency, and speed, all while maintaining quality.
Mm-hmm. And I see the more effective gene therapy appears to be and seems to be in the clinic, more and more larger, well-established companies will become involved. And I would like to see those partnerships develop along with the regulatory agencies to address more mainstream indications and again, a larger patient population. This is a very exciting technology, and it has a lot of there's a lot of hope for previously uncurable diseases to be treated. So I'd love to see that partnership grow with the agencies and the companies working on these technologies.
Right. Okay. So Heidi, Marc, firstly, thank you very much for taking the time to join me today and to share with me some of your ideas. Thank you also for being truly pioneers in this fantastic modality, where this, to your point, Heidi, where, we're offering cures for children who perhaps historically had no hope, no future. So really, really exciting, super cool. And for those of our viewers who are watching us, that's it for this edition of All In with Bulpin. Join us next time when we're going to be discussing yet another topic in the exciting world of biopharmaceuticals. Take care, and we'll see you soon.